Hello everyone, my name is Joseph McCreary and I'm a second year master's student under Dr. George Hong in the Department of Biochemistry and Microbiology at CHS. Today I'm going to talk about an antimicrobial protein called the Bacteriocin that targets C. diff. I'm going to start with a quick background on C. diff. It is a gram-positive, rod-shaped bacterium. It is anaerobic and spore-forming. Transmission is through the fecal-oral route. And risk factors include antibiotic exposure, old age, a recent hospital stay, weakened immune system, and a previous history of C. diff. Treatment is highly dependent on the severity of disease, which ranges from asymptomatic carriers to pseudomembrane colitis and death. C. diff can resist most antibiotics and are currently only treated with the two antibiotics metronidazole and vancomycin. Both of these are non-specific to C. diff and have the side effects of killing the normal gut microbiota. An intact gut microbiota actually helps to prevent C. diff infections. This lack of specific treatment options perpetuates the problem of infection and can lead to a relapse in disease. Clostridium butyrhydum, a non-pathogenic probiotic, has been shown to produce a highly specific antimicrobial product called a bacteriocin. A bacteriocin is a peptide or protein that gets produced by closely related species of bacteria to outcompete one another. This bacteriocin, produced by Clostridium butyrhydum, targets C. diff. Previously, the gene encoding for this bacteriocin, CBMB, was cloned into E. coli and purified as a recombinant protein. The recombinant CBMB has shown to exhibit potent activities on multiple strains of C. diff. It has also been shown that CBMB consists of two parts, a long non-catalytic domain located at the N-terminus and a small mature fragment that possesses the activity. For my project, I want to continue to characterize CBMB. I did this by performing two tests, a growth curve analysis and a disk diffusion test. To get to these tests, I followed the following steps. I transformed BL21 E. coli with the plasmid containing CBMB, our vector being a PET21B. I then followed an induction protocol. From there, I collected the cell pellet, sonicated to lysocellus, and purified using a nickel resin column. Using the His tag that was added by our vector. I further purified by FPLC to collect just a small active portion of CBMB. With the purified protein, I performed a growth curve analysis over 8 hours. I inoculated BHIS broth with C. diff lab strain called R20291, and each broth it either contained 6 or 3 micrograms per milliliter of vancomycin, or 12 or 6 micrograms per milliliter of CBMB. There was also a control that had no antimicrobial products. Looking at figure A, you can see that the control grew quite nicely over the 8 hours, while as for the 4 inoculations containing antimicrobial products, those failed to grow. This is better visualized if you look at figure B, where you can see that there is a very significant decrease in OD compared to the control within just the first hour of growth. Looking at figure C, you can see this trend kept through the whole 8 hours, where there's a significant decrease in OD between treatments when compared to the control. It is also important to highlight that CBMB performed similar to vancomycin at similar concentrations for the 8 hours. Next, we wish to better visualize the antimicrobial effects of CBMB in comparison to vancomycin. To do this, we used a heavily modified disk diffusion assay. We chose to do this because the nature of C. diff being anaerobic makes it difficult to do a spot-lawn assay. Its anaerobic properties does not give us a whole lot of time to allow antimicrobial products to dry on the agar plate before C. diff starts dying or forming spores. Thus, most of the images tend to look like the one furthest to the right. To our surprise, though, the assay was unsuccessful in what we wish to accomplish. We can see that both strains, R20291 and 630, Vancomycin had a clear zone of inhibition, while CBNB only showed a very slight clearing for R20291 at 100 micrograms per milliliter. I think this may be due to CBNB not being able to easily diffuse out of the disc as vancomycin can, or it might be due to some other factor that I'm not aware of. Lastly, I want to talk about some in silico work that I performed on the structure of CBNB. A paper published last year in Nature introduced the newest iteration of AlphaFold. AlphaFold is a machine learning AI that can, with high accuracy, predict the structure of proteins. Source code was made available for free, and using it, I was able to predict the structure of the full unbroken CBNB protein. This is seen on the left with the two domains highlighted in red and blue. Then separately, I predicted the structures of both the active portion and the inactive long N-terminal domain. These colors correspond to the first image. I then overlaid them to see if there was any structural differences. This is the image the furthest to the right. What this shows me is that the active portion of CBNB is not reliant on the inactive portion to fold correctly. This points to the small active portion being able to be cloned into E. coli and produced similarly to what we're doing with the current full CBNB fragment. It allowed us to also predict 4 more structures of peptides, each containing the sequence of 4 of the alpha helixes in the active CBNB fragment. Each one is named and the structures are predicted. Then they are overlaid with the active CBNB fragment. We then had these peptides commercially synthesized. Our hopes is that these will allow us to determine the active domain of the CBNB fragment. So here's a quick summary of my poster and the future directions I wish to take this project. CBNB has a very significant decrease in the amount of bacterial growth when compared to the control with no treatment. CBNB also has activity that is very similar to vancomycin at similar concentrations. Structure predictions using AlphaFold 2 point to the non-active internal domain not influencing the folding of the active CBNB fragment. And then also with the structure predictions, we were able to synthesize 4 peptides that we predict should have antimicrobial activity against C. diff. Where I'd like to take this is, given the structure predictions, we believe that it's possible to clone just a small active portion of CBNB into E. coli, and this is something I'm currently working on. We also wish to test our 4 peptides in the same fashion that we're currently testing all our the regular CBNB fragment to see if we can determine that active domain. I want to thank everybody for listening, and I hope you all have a nice day.